So this session is now about the hardware and software requirements that we need to consider when we want to establish an ECoG Stereo-EEG-based brain-computer interface. And yeah, let's check already what are the general requirements. So what we want to establish is a BCI that reads the, sorry, I have an echo here. Okay, better. That reads the signals directly from the brain, here from the electrodes, undergoes a pre-processing and a feature extraction step. And based on the biomarker that we extracted, we want to make a decision. So based on classification or detection. And then we either get feedback as a user. So this can be utilized, for example, for neuroplasticity or rehabilitation or diagnostic purposes. And the other solution is that we have a control signal when we want to control an external device, like a humanoid robot, prosthesis, wheelchairs, or even a muscle stimulator like a FES. And the definition is that we have a system that controls a device without the brain's normal output pathway, and also it must be in real time, so that we have explicit bounded responses in time constraint. Otherwise, we will experience a failure of the system. So those two requirements are essential for the setup, which we have to consider now when we want to establish the system. So where do we get the signals from? Basically directly from the brain. So we can either utilize epidural or subdural ECoG. So here we have the dura mater, and if we are connected directly to the gray matter, to the cortex, then this is called subdural electrocorticography. And other ways to obtain direct brain signals are from local field potentials when we penetrate the gray matter. Or we can also record, for example, action potentials or spike from very small tetrodes that are penetrating the gray matter. And another way with more bigger sites like macro electrodes called stereo-EEG from stereotactic surgery. We have here the depth electrodes also penetrating the gray and the white matter. So we have recording sites from deeper brain structures as well. And in contrast to the surface EEG, the intracranial signals have, if we're talking about macro electrodes like stereo-EEG and ECoG here in green and blue, they have a much stronger signal amplitude compared to surface EEG. And also the frequency range that we can utilize. So the damping is much less and the frequency range is higher. So we can easily maintain signals and utilize biomarkers from higher frequency components up to 200 Hertz or even higher. So some groups already showed that we can utilize broadband features up to 1000 Hertz with electrocorticography. In contrast, usually the EEG biomarker stops at 40 Hertz, which is just below the mains frequency of 50 or 60 Hertz. And the spatial resolution is, of course, lower with about a centimeter conductive diameter, and then a spacing of about usually 2 centimeters. So here we have much smaller diameters, like about 2 millimeters, like almost a fifth or a tenth of it. And then usually for sEEG and for ECoG, we have similar diameters and exposed area. The main difference is that with the electrocorticography, we are just in direct contact with the cortex, the gray matter, and with sEEG leads, we usually have white and gray matter contacts mixed. So these are examples. So here we have hybrid electrodes that can be used with gel or dry. And also here we have an example for a 64-channel ECoG grid that will be placed when we have a craniotomy. So then the skull is removed, and the skull and the dura mater is opened, and then underneath the dura, usually the grid is either exposing the open craniotomy area, or it is slightly moved underneath the dura. In contrast, the stereo-EEG electrodes, they're actually implanted via screw holes, so the skull is not removed for the implantation, and there are only screw holes during a stereoscopic or stereotactic surgery where the shafts are then penetrated into the brain directly. So here are examples for electrode connections. This is one of the major electrode manufacturers in the United States, g.Tec, and here we have a standard grid clinically used, usually with 4 by 5, so 20 electrodes in total, with 1 centimeter spacing, and here the exposed diameter is 2.3 millimeters, and that is a clinically widely used grid, mainly for the purpose of epilepsy monitoring and seizure localization for epilepsy treatment. And the second use case is an intraoperative monitoring, for example, during awake glioma surgeries when the tumor has to be removed in an awake surgery use case. And then we have here usually the pigtail, so the connection is completely passive. There is no preamplification in the electrodes. Then we have the connection cable with the connector block, where the pigtail is irritated, and then the normal output are the 1.5 millimeter safety touch-proof connectors. And with this, we can connect them directly to a passive head box. So this is our head box that we usually use with 64 electrode connections and a ground socket. So in total, 65 electrodes can be connected to this box, and then this goes to the biosignal amplifier. The alternative would also to directly connect the electrode with a multi-pin connector. So here, for example, this is a quick connector with an auto system that is interfacing the g.Tec grids, and with this we actually save the time to manually connect each individual electrode. So you have to imagine if you have a full system of 256 channel or even higher, then this is a very tedious and long process, and you must not do any mistake here, especially for diagnostic purposes. If one electrode is mixed, then the whole localization pipeline can be destroyed. So this is a very difficult and dangerous procedure. This interface system also is available for Cortec, a German manufacturer, and of course we can customize that for any other. So now when we record from those electrodes with the biosignal amplifier, then we get a voltage signal for a recording side against ground. And this voltage represents now the brain waves, and we can look at this in terms of a spectrogram. And now when we look at the spectrum of rest, we can see this 1/f behavior. So the higher the frequency, the higher the damping effect of our spectral power. And now when we perform any task, so any behavioral change, then like in this hand movement, we can see that there is a change in the brain signals. And now what we can see, and this is different from the surface EEG, so here in the lower frequency components, we can see still the same phenomenon as we saw it in the surface EEG, the oscillatory suppression. But beyond main frequency here, you can see this peak of the main frequencies. We can see now a broadband lifted band power. And this starts usually here around 60 Hertz. And we can see that up to 1000 Hertz basically until the noise flow of the amplifier. And this is now very interesting because when we compare the biomarker for the low frequency band suppression and the high frequency band augmentation or enhancement, then we can see that this high frequency band activation is very focused on the cortex. So only a few electrodes show this behavior during this movement task. Whereas the low frequency band suppression is widely spread over the whole cortex. So many of the electrodes here on the motor cortex are actually suppressed in a low frequency band, but only one electrode is finally catching all this broadband activation. And in some connected studies, we can actually see that this broadband activation is related to the neural firing rate, and also co-localizes with a bold response in fMRI. So this is a direct marker actually for the neuron firing rate underneath this cortex, whereas the low frequency suppression can be more considered as a conditioning of the cortex so that it's easier excitable, and this can be related to an anticipation effect of the upcoming task. And this phenomenon is now known since the last more than 25 years. So Nathan Groen showed this phenomenon in a study in 1998. And since then, many groups tried to utilize this broadband activation as a biomarker also for brain-computer interfaces. So here is an example 18 years ago from the University of Washington, where you can see a two-dimensional cursor control. So the two dimensions are basically created by the, in one direction, the motor, hand motor sites on the hand motor cortex, and in the other direction on the oral motor cortex. And yeah, when we take the features from the oral motor cortex, we can also now look at an example from a brain-computer interface that was established by Gerwin Schalk in BCI 2000. So here the subject is now playing Doom, the video game, with a joystick with the hands directly, but the shooting is solely triggered by the oral motor cortex. And by showing this demonstration, Gerwin also got a lot of attraction at the US Army. And of course, this was then for him also an opener for further research. Another example is when we want to interact, for example, through robotic devices. So here we can see an experiment that we set up together within a joint collaboration with France and Japan University, where here in a Japanese patient, actually a humanoid robot was controlled by using only motor imagery band power features from ECoG grids. And here the humanoid robot received high-level commands for grasping the can, and the patient here just imagined to grasp the can, and after several trials the robot was able to do it based on the trigger from the patient. So in order to get to this stage, what do we have to consider? And when we design an ECoG experiment, we have to understand that this is relying on many factors, especially the clinical environment. So usually it's not possible to simply decide that we want to do an experiment. So we have to get access to actual patients. So the majority of the patients that are contributing to BCI research are epilepsy patients. Sometimes also ALS patients are volunteering in implanting electrodes, but those are quite rare cases. So usually when we have the clinical opportunity and the need for an implantation, then in some occasions it's also possible to reserve time for research experiments, but this time is very limited. That means that the experiment has to be designed quite well beforehand. So what has to be defined is actually, and that's mandatory, kind of a task protocol. So we have to have a hypothesis, and we have to understand what the patient has to do during the recording so that we can analyze it and get useful biomarkers for our experiment. Then, of course, we have the channel assignment. So I previously explained that we have to connect each individual electrode to the amplifier. And it's very important that this channel assignment is correct. And usually in a hospital environment where the patient is already connected to a, for example, monitoring system for epilepsy localization, then we have to coordinate that with the EEG tech in the hospital so that we have a common channel assignment and a common lookup table for all the connection of the electrodes. Then we need a control computer that is analyzing the signals and showing us the raw data. So this control computer is connected, in this case via USB, to our g.Amp amplifier, and then the amplifier can be connected up to 256 electrodes. Of course, we can also stack it if there is the need for more electrodes, but the default configuration usually goes up to 256 electrodes. And then we need a processing platform for the actual recording and feature extraction for online processing. So for that, we usually use our g.Hysys in Simulink. So we can save the data on the PC for post-processing, but we can also, at the time of the recording, visualize it, filter it, and decide whether or not the feature, for example, can be classified. And then we also have to think of how to analyze it later on, so as a post-processing pipeline. And here we basically use MATLAB, and for visualization and filtering purposes, the GBS Analyze toolbox that is connected to MATLAB. And so this is the minimum requirement, and after this step, we can now establish, of course, a more sophisticated experiment by a closed-loop neuromodulation setup. So here we have the possibility to connect a stimulator, a cortical stimulator, the eStim Pro, and a switching unit for stimulating and recording, the eStim Pro switching unit. And then the switching unit is, of course, the interface to the electrodes and can route the amplifier and the stimulator for sensing and stimulating. Another improvement is now the topographical visualization. So we can actually translate the channel assignment table into 2D topographical visualization so that we understand which electrodes actually belong to a grid or a shaft, and also which electrodes are in a certain geometry compared to each other. So this already helps to understand if biomarkers are meaningful and if common activation is actually explained by the geometry. This can be further improved by utilizing medical imaging to localize the sensors directly in the patient's brain. And for that purpose, we have a pre-surgical CT, a pre-surgical MRI, sorry, and a post-surgical CT, and optionally also fMRI and tractography DTI imaging can be used. But in order to understand where in the brain our sensors are located, so that we can, for example, decide based on the region of interest which sensors are useful, we need to have at least an MRI and CT scan. And now here we can see that not only the geometry of the grid is now visible, but also the location of the electrode directly on the cortex. And the complete system looks like this. So for recording only, we have here now the head box with the connection cable to the amplifier, the processing PC here with software CortiQ running to show the raw data and the brain activation by means of bubbles, and a patient monitor that is also usually mandatory to instruct the patient what is actually the task, or to present, for example, visual stimuli to the patient. And the more sophisticated setup is actually then with the switching unit and the eStim Pro, so that we can then at the time of the recording decide based on the features if we need a stimulation, where we need the stimulation, and when we want to stimulate and switch from the recording to the stimulator. So this procedure is very quick, so it just takes between 3 to 5 milliseconds to make a decision to switch or record. So the majority of the actual delay comes from the signal processing. And this, of course, just depends on the biomarker. So how do we connect this now? So we have to consider that the patient is in a hospital environment, and we have here the patient on a patient's bed, and then the electrodes implanted, the head boxes next to the patient, and then the connection to the amplifier and the switching unit and the control computer. So this looks like this. So the front interface is that we have the selection of the grids and shafts. We connect them directly or via the head box to the electrode sockets, and then propagate the signal to the amplifier, and also the stimulation current comes from the stimulator into the switching unit, and here the decision is made what to record and what to stimulate. And on the backside, there can be more cables, especially when we need to propagate triggers through the devices and trigger them externally. So it's possible, for example, to read external behavioral triggers. So for example, when there is a microphone threshold, when there is a push button trigger information, we can read this TTL information into the amplifier, and by that synchronize the actual protocol, the external protocol, with the raw data. But we can also, based, for example, on an external decision maker like an Arduino or any digital output that is sent, we can activate the stimulator and the switching unit to route and stimulate, and then we get the stimulation trigger back into the amplifier. So when we now actually connect the amplifier to the patient, there are a few considerations that we have to follow to get the proper signal quality. And in order to understand that, let's have a look at an interoperative setup, because this is usually a very bad environment for obtaining a signal, but it's mandatory to have EEG signal in the operating room, of course, for diagnostic purposes. And that's why we start here to get a good signal. This is an example how an operating theater can look like. So here's the patient getting prepared for the surgery. We have stacked all the devices that we need for recording and for stimulating. Here are some head boxes, and then there's the stacked switching unit with the amplifier. So there are two ways to run them. So one is with the mains power supply. So we have actually the mains power outlet in the wall, and then we can connect all the power cables to the control computer and the amplifier. But it's also possible to run everything fully on battery. And we have to understand what is now the best setup. And in order to record the signal, we also have to select the ground and the reference electrode. So now imagine here there is maybe the seizure onset zone in this montage. We have to understand which ground and which reference electrodes are now useful for a good recording. So usually the ground and the reference electrode have to be distant from the region of interest. This can be for BCI purposes, but also for diagnostic purposes. So here now in yellow and blue, we have the reference and the ground far away from the region of interest. So the ground electrode is the most important electrode. If the ground electrode is broken, then we don't get any signal. And also, if, for example, the ground electrode is from a different material, so it could be a subdermal needle, then we have to be very careful if the recording electrodes are also from a different material and in a different tissue, then these two electrodes can create a very high polarization voltage. So one example would be a subdermal needle from stainless steel and an implanted recording electrode from platinum iridium. So here we have to be very careful, and if this polarization voltage is very high, then the amplifier can saturate. So there are a few ways to mitigate that. So one is, for example, by adding the switching unit or an analog high pass, we can remove the DC drift or the polarization voltage, the DC offset, and by that have a zero mean signal again. And with that signal, we cannot saturate again the amplifier, at least from the ground electrode. Usually we like to have the best similar overlapping characteristics between the electrodes. So an intracranial ground electrode, this could be, for example, a grounding strip or a a white matter electrode in a shaft, or sometimes it could be, for example, a skull-based ground. It could also be a ground strip that is dura-facing. So there are multiple ways to define the ground, but important is that it's actually in contact with, so that it's of the same metal, and that it's preferably in contact with a similar tissue as the recording electrodes. So now when we compare the signal quality between the epilepsy monitoring unit here on the left side, where devices are turned off, and in the operating room when devices are turned on, we can actually see that in the operating room, the interference, the contamination with mains frequency of 50 or 60 Hertz is much more severe or higher compared to the clean setup. So now we have to understand how we can minimize the interference when other devices are turned on. So of course, the first step is to turn off everything that is not necessary. But if this is not possible, we also have to make sure that there is a proper connection to Earth. So here, when we now establish a ref electrode, so here, this is an unreferenced signal, no ref is used. That's why we can see that. So the first step is to use a proper ref electrode, and by that remove the common signals. And this common mode suppression actually will remove the 50 or 60 Hertz interference tremendously. So here only this ground minus ref channel here tells us what was actually the original interference, but we don't see that in the reference signals anymore. However, this is only valid as long as we have a proper Earth connection, either with the amplifier or with the PC. And so in order, for example, if now the PC has no Earth connection anymore, then this can be tremendously increase, or this can tremendously increase the interference here that we see. And one solution is that we make sure that the notebook is either always is always powered with power supply that actually has a good Earth connection. So we have to be sure. But sometimes in electrical installations, it can happen that there's actually no protective Earth in the socket, and we have a potential equalization pin in the wall. So now we can utilize, for example, any Earth connection in the room, like this PE pin. And when we now connect the USB to this Earth, then we can see that the interference is basically minimized. So this is very important that we have a proper setup. So going back to the ground electrode, we have to understand that the impedance of the ground is also a crucial factor. So how do we know if the impedance is good? So we can make an impedance check, either with the G.high amp or with the switching unit, and we can see basically if the impedance is measured from all the from each electrode against ground, that the majority of the electrodes will have a relatively high impedance. So here indicated in yellow. And when we change now the ground electrode to a good contact channel or a good contact electrode, then the majority of the electrode goes to green, which is basically below 5 kilo ohms for intracranial electrodes. So this is a common impedance range, and we should really try to get a recording within this range. So of course, some electrodes are simply not in good contact, like this here, 75. But if the majority is not in good contact, this is a very strong indicator that we selected a very bad ground channel. And if the ground channel is good, that will solve many problems from the very beginning. So here you can see when we now record against this channel 7, which is actually with a high impedance, then here we can see big interference, and also here we can see a big interference in this bottom channel, where the interference basically diminished for a low impedance ground channel. So now that we have a good signal quality and we get actually a meaningful signal out of the amplifier, we now have to define a pipeline how to process and record the signal. So for intracranial recordings, a sampling rate starting from around 1200 Hertz makes sense. So with this, we can cover the majority of evoked potentials and oscillatory and broadband phenomena. If we want to record even single neuron electrodes, then we of course have to go higher, and we can go up to 38.4 kilohertz. And the input range of the amplifier is 340 millivolts with a 24-bit analog digital converter. And the characteristic of the g.Amp is that it's DC coupled, so we can even measure DC drifts with it, but we have to take into account the electrode polarization voltage. So if we want to avoid that, then we can also take the AC coupled input stage of the eStim Pro switching unit with a high pass cutoff frequency of 0.16 Hertz, which is still relatively low, so that we don't bias evoked potentials if we want to analyze the waveform of the evoked potentials. But it's already good enough to get rid of the polarization voltage. And then when we run a real-time experiment, we also have to define an update rate of our actional signal processing. So it doesn't really make sense to update every millisecond for a 1200 Hertz sampling. So it makes sense, for example, that we collect a frame of samples and then analyze the whole frame, for example, every 7 milliseconds. And so then we run at 150 Hertz. And here you can see an example pipeline for a BCI that is actually taking the signals, doing spatial filtering to reduce the feature dimensionality, then here extracting the signals of interest in terms of a temporal filter, and then the band power estimation, the classification, and the feedback, for example here, direct feedback to the user or control of robotic hands. Then we have to define a protocol, what the patient has to do. So for that purpose, usually we have a tree structure with tasks. So we can have very deep structure and complex combinations if we want to have randomized blocks of images or audio files, but it can also be a simple instruction with a very flat tree. So here, for example, this could be a caption with an instruction, do a movement, but it can also be a hierarchy with many here leaves of individual images. So the complexity can be designed by the designer of the experiment. And then once this tree structure is established as an XML file, we can load that in g.Sys in Simulink, and now open the paradigm window. So here you can see the operator window with the time until the experiment has ended, the status of the protocol, and then there's also the patient window that we want to show on the patient screen. I will just show it here for demonstration purposes on the same. And then we can see the raw data visualization scope, and we can now acquire the data. So here you can see this is the amplifier block for an actual online experiment. And here this is a file replay block when we want to replay previously sessions and analyze them retrospectively. So this is also very nice to simulate real-time experiments so that we can first test and be sure that everything is correct. Here's the presenter block, and we get the task ID and the group ID as channels. So this is the trigger information. And now here you can see this is a visual presentation paradigm where we can see different objects, digits, kanji characters in color and grayscale. And we can see the ECoG data that is now here recorded at the time of the presentation. And in the bottom channels, we can also see the task information about here the event ID of the picture, and in the bottom, the group ID, how the pictures can be grouped, for example, into category face, category kanji, or category black, and others. So now we have recorded the brain signals synchronized with the task. And the next step is to understand if there are meaningful features or biomarkers in the data that can be task related. And for this purpose, we now offline process the recorded data, and we can launch our offline processing toolbox GBS Analyze in MATLAB, and then load the EEG data file. So the g.Estim saves the data in MATLAB format. That means we get the MAT file, and then we can load the MAT file directly in MATLAB. We have to specify the sampling rate and the paradigm information, which is basically saved together with the raw data. And now you can see that the data is actually with a DC offset. So the first step that we have to do is to apply a visualization high pass. And now we can see the ringing after the step function. And now we want to properly adjust the visualization. So we here show now 100 channels over 20 seconds. And a useful signal amplitude is about 200 microvolts in each direction. So once this is scaled, we can see a nice and clean EEG raw data plot. So the first analysis that we perform is a signal quality check. And what we want to understand is which electrodes show outliers, so transient events, which electrodes show high correlation, crosstalk, which is an indicator for open leads, and also which electrodes have a very high mains frequency interference. This is an indicator for a high impedance or bad contact. So this takes a few moments, and then we get for every second in the raw data an annotation whether or not this is a good epoch. And so usually if more than 10 to 30%, depending on the task of the data, is contaminated, the channels should be not considered for the analysis at all. If we only have transient artifacts, we can consider that we exclude some of the contaminated trials, but still include the channel. And if a channel is generally good, then of course we can keep them. So here you can see the majority of the signals look clean, and at the beginning, in the ringing settling artifact of the filter, we can see a lot of selected bad epochs. And now when we scroll through the data, then we identified one channel in red that is really bad, that is just floating around with huge amplitudes. So we will not consider this channel, but the rest can be used after the filter stabilized. So now as a next step, we would like to perform re-referencing. So a very good start point is a common average reference. So usually for grid electrodes, this is the default way to re-reference the data. For stereo-EEG, it's also possible to use a common average reference, but for some detailed analysis, it can be also preferable to have, for example, one local average reference per shaft, or even bipolar recordings of neighboring electrodes. So now, given our protocol, so we had a visual presentation task. That means that the trials were relatively short, and the presentation time of the images was only a few hundred milliseconds. So we can take a very short presentation baseline of 500 milliseconds and about 1000 milliseconds after the presentation onset. And then we have to load the marker information from the paradigm info that is created by the presenter. And here we have the different classes now that we want to analyze. So one is phase, another one is kanji, and another one is just rest or no change of the black screen. And now we can see on the top how the trials have been annotated. And now we will look at the spectral changes for each of the classes. So for that purpose, we will create time frequency maps or ERD maps between 5 and 145 Hertz. And we will select, of course, all the electrodes. And our baseline interval is now selected as 200 to 500 milliseconds after the ECoG start, and our trigger of the marker was at 500 milliseconds because of the pre-trigger time that we selected. So 500 is the beginning of the flashing, and we will just end our reference period when the flashing started. So now it's calculating the time frequency maps for 150 channels. And when we get that, we can, or 145, and when we get that now, we can see here very tiny these boxes where we see from 0 to 145 Hertz and the length of our epochs that we defined beforehand. So this is a bit difficult to understand. What we already can see is there are some blue blobs in some of the boxes. This is the high gamma activation. So there are some electrodes that are totally off and some electrodes that are now very much reacting and showing brain activation for the visual perception. What we don't know is what is now the detail of the TF map and also where it happens. So now we want to add the topographical information, and for that purpose we have to create a 2D montage at least. So if we don't have any information, for example, from MRI or CT, we can basically use the information from the grids that we used for the recording. And for that purpose, we now have to define a background image. This can be a topographical image of the brain on the brain, so that this topography fits our recording. So we can utilize here the Cortec montage creator to do that, and then use the topography also in MATLAB to make ensembles of the electrodes and better interpret the biomarkers, how they relate to each other and how we can utilize them, and if they are, for example, in a region of interest or not. So now when we created the montage and loaded for the ERD plots, we can already see that the resolution is much better when we show the activation grid-wise. And so here on the top left, you can see a 60-channel grid, but still numbering is not according to the grid shape. And on the bottom right, you can see now the grid shape is also taken into account. And we can now understand that there are actually some regions of the cortex where we have a grouped activation with a hotspot of basically 5 to 6 electrodes and a bit of minor activation surrounding of them. So now if this is a meaningful activation and a task-related biomarker that we want to utilize for example for a brain-computer interface, we can now calibrate a classifier based on this data that we have already stored. So we need the task related biomarker annotated with the task labels, and then can extract a classifier to discriminate, for example, between phases, kanjis, and rest. So to do that, we again have to load the data into our offline analysis toolbox to g.Base Analyze. We have to properly scale again. Okay, this looks already quite good. So here we have now 10 seconds of ECoG data. Again, the majority of the channels is pretty clean. Here there's one channel with interference. On the bottom there are also 4 channels with high interference, and on the very bottom we can see the task trigger information. So now that we have already an understanding of our biomarker distribution, we can already utilize a filter band that is dedicated for the BCI experiment. So in this case, because it was a 50 Hertz mains frequency, we are now filtering between 110 and 140 Hertz. And this bandpass is already a good filter band for a high gamma band power feature. So we will call that high gamma activation, HGA. And so for the calibration here, we can use an anti-causal filter to avoid any ringing effect. And then on the right side in the MATLAB command line window, you can actually see that all the steps that we perform in the data editor are also visible as a scriptable code in the command line. So we can basically copy and paste the code from the command line into a MATLAB script and then generalize the script, for example, for multiple subjects and for a variable amount of channels, and for, of course, variable frequency bands and so on. So now we have to scale down to 10 microvolts because we are now in the high gamma frequency band. And what we can see, it's a quite narrow band. That's why we have such kind of oscillation. But we can now see when high gamma events occur in the brain. And you can see that they are bursting occasionally, depending on the channel and the time. And now we are again chopping the data into the trials of the visual perception. So now we can see the high gamma activation of the filtered raw data for different trials. Again, we are loading the paradigm information with the event markers. So for phase, kanji, and rest. So we have the annotation again in the data editor. And now, in order to make a real-time BCI actually capable of real-time processing and not being too demanding for the capacities of the PC, and also to get the best features, we are now applying a common special pattern analysis. This is a sort of principal component analysis where the signals of 2 classes are basically spatially filtered to maximize the variance for one class and minimize for the other. And this is then solved with an eigenvalue problem where we get for the amount of channels eigenvalues, and for each eigenvalue we get an eigenvector, which is a linear combination or spatial filter. And we will take the two filters that are maximizing the variance for class phase, and then we will also take the two filters that are actually the two last eigenvectors, and those are maximizing also for class 2. So exactly the opposite. And we repeat this for also rest against phase and kanji against rest, so that we have a free class problem. And then we have the features, the feature channels, so the spatially filtered channels here with a certain amount. So for each class, basically we get 4 channels. And these channels are now representative for different tasks. So you can already see that in the first trial, only one channel responds to kanji. And now we are also normalizing the signal. So after the bandpass filtering, we have the spatial filtering. After the spatial filtering, we have the band power estimation here with a variance estimator. And after the band power estimation, we have the log transformation to ensure that we have a Gaussian distribution of our band power features. And finally, we will create the linear classifier that can discriminate between phases and kanjis. So now when we get the trials with the features for phases and the trials for features with kanjis, and we have the labels, then we can perform a cross-validation to see how good is the performance of the classifier. And as we can see already with the eye of the individual feature channels, that the feature transitions are unique for each trial and each channel. Basically, we get a perfect accuracy between half a second to 700 milliseconds after the cue. And now we want to utilize this period for online detection. So we will select the filter, the LDE, the weights, the classifier weights of the features, and take the weights from those times, so between 500 to 700 milliseconds after the presentation onset. And so now we want to see how this biomarker output with the classifier decision looks when we do an online classification. So we can feed now a validation dataset into the online processing pipeline. We can select the classifier here after 625 milliseconds after the cue. And then we have here the bandpass filter in green, and in blue the common special patterns, the spatial filters, and then the normalization of the band power. So this is now a MATLAB figure that gives us feedback of the classifier output. This is a feature scope that shows us how the band power feature channels look like. And now you can see with the green shaded areas and in this queue window, if the actual protocol and the BCI output match. And in dark green you have a phase, and in bright green you have a kanji. And if it's white, it's rest. And now you can see in the queue if there is a kanji. And in the left feature scope, you can see that it's bright green. So now we have a queue and a phase, and here in dark green the phase output. So this paradigm feedback window is linked to the feature scope. So whenever it's green, there will be an output, and we will see if this corresponds to the cue here. And now we can actually go to the patient and perform the experiment in real time with untrained images of faces and kanjis. And we can see now in the center the feedback monitor of the BCI, and it reacts within half a second to the perception of the patient directly obtained from the brain, and it can discriminate between a face and a kanji. It also works for the patient's face himself, foreign faces, and interestingly, for the doctor who was always in the field of view, it's not as strong. We can use the same pipeline also, for example, for hand motor control. So here we use the same pipeline with high gamma band power and CSP features for propagating the brain signals directly to an arm or hand robot that mimics then the actual movement in real time. And now we want to go to the next improvement. So now we have basically the pipeline to get good biomarkers, to have a decision to send it to the outside. And now we want to understand where this happens in the brain. And for this, actually, we need now to also consider medical imaging as the pre-surgical MRI and the post-surgical CT. And we need basically to segment the brain first. Our usual pipeline that we utilize is FreeSurfer or FastSurfer to get the cortical segmentation, the deep structure information, the ASEC, and the cortex pacillation also, so that we also already have the anatomical landmarks. And of course, any other segmentation pipeline can be utilized, like SPM or 3D slicer, but we usually use FreeSurfer for that purpose. And we have a 3D montage creator that can be actually connected to CortiQ and HISUS for visualization. And then we can import the CT scan and the segmented brain to localize the sensors. So let's have a look how this works. So there are multiple workflows, and now we have a workflow where we want to localize ECoG grids on the brain. So we can see here the pacillation that comes from FreeSurfer. We have the slice information of the MRI, and now the first step is to compute a hull so that we can actually project the electrodes onto the hull so that they're in direct contact with the cortex. This is necessary because actually the electrodes are inside the skull when we implant them, and they squeeze the brain a bit so that it looks like and appears that the electrodes are inside the brain, but in fact they are on the cortex. So we need to correct this brain shift. First, we also have to co-register the CT scan and the MRI, so we can do that based on the skull of both images. And now we have the CT in the same space as the MRI and the brain model, the segmented brain. And now we can actually get rid of the cable artifacts with a cropping mode. And we can see the electrode artifacts already very nicely. So as clean as possible, and after the cropping, then we can automatically assign the electrodes based on the blob artifacts of the CT. And some of them are grown together. That means that we have to correct the positions and remove duplicates or add new electrodes. But the majority should be okay. And then here you can see now the electrodes appearing in the brain with the brain shift, and we have to correct that. So now we project the electrodes onto the actual surface. And now we can see here the electrodes on the cortex. And now we can assign them basically to a region of interest in 3D according to the anatomical landmarks. Okay. The same thing can be also done with stereo-EEG electrodes. So we can place them either in a region of interest if we don't have the CT information available. But if we have the CT information available, then we can do similar to the ECoG co-registration and localization. First, remove the cable artifacts. So here on the left side, we have also a history of the cropping. So we can revert that if we make any mistake. And now we can see already how the shafts are co-registered with the segmented brain and with the MRI and the CT. And now the remaining segmented shafts are used to automatically assign the shafts from the Corteq Electrode database. So here we can now see in white the artifacts of the CT, and in blue the virtual shafts. So they're automatically placed in the orientation of the CT artifacts. And now we can select the individual shafts and modify them so that they perfectly fit the geometry of the electrode artifacts. So for stereo-EEG, we don't have to compensate for any brain shift because they're penetrating the brain, but not actually changing the brain location or squeezing the brain. That's why we can utilize the original position of the CT. And now we can snap the correct shaft that is actually implanted from the database into the CT artifact and correct only along the trajectory of the artifact the position of the electrodes and snap them to the center of the CT. So now with this mechanism, we can very quickly and easily define the electrode positions for the recording. And now basically we can understand where in the brain the activity happens exactly. So now here, if we remember the robotic hand that was following, so now we can look in a patient here with stereo-EEG implanted when we perform a grasping task where the actual activation happens. And here there are some in the Rolandic cortex, actually in a deeper medial structure. We can see here in the slice the activation of this grasping. Another aspect that we can consider is now, for example, for the visual perception, that we can understand based on the pacillation where the activation happens in terms, for example, of the fusiform gyrus, the inferior temporal gyrus. And if we now want to understand, for example, the visual fusiform face area, then we can see here that this visual percept, when we show faces, is only firing for face percepts, but not for any other visual category. So this is very specific for faces, and now we can see that this is actually on the fusiform gyrus. And we can also visualize now the time frequency maps directly on the fusiform gyrus, and we can see, for example, here the broadband activation over the whole fusiform gyrus for face percepts, but only on more here posterior sides for actually the kanji. Another advantage is now that we have, for example, regions of interest and the sensor localization, that we can merge them on a template brain, for example, the MNI 152. And by that we can see now in group analysis, for example, the distribution and localization of the electrodes and their activation. Another advantage is that we now can co-localize electrode activation with fMRI activity. So once we have the activation information, we can actually show which electrodes are related to bold response, which electrodes are related to high gamma features, and which are actually showing, for example, symptoms when we stimulate them electrically. And this allows us now to perform a functional mapping on top of the BCI features, for example. And this can be further used for diagnostic purposes. And later in the spring school, next week, we also have a dedicated session only for the functional mapping, and you're also invited to join this one as well. Another aspect is in the epilepsy monitoring to utilize, for example, 3D visualizations to localize seizures. And if we now want to understand how electrodes are interconnected with the brain, then we can also show that basically. Sorry, now my presentation just stopped. We can also show that here, basically, to stimulate, record in real time at the electrodes that we want. Okay, so I hope that this basically gave you an overview what to consider, what is possible to really establish an invasive BCI platform, and what has to be taken into account to make it happen. I think that we're already out of time. So thank you for listening, and yeah, I hope you will have successful BCI.
Thank you very much, Capi. Alberto is asking how comes that the stereo-EEG is penetrating, the scalp and the brain tissue, that you don't get side effects?
Well, I think there is actually a big debate about whether the penetration of the stereo-EEG is influencing the brain or not. So in terms of symptoms, it's not really it's not known yet that the sEEG shafts are causing obvious brain damage. So the shafts are small enough that they're not actually causing too many direct problems. It's important that you're not, of course, hurting any vessel in the implantation stage. So a bleeding, of course, is very dangerous. But other than this, the shafts appear to be quite safe for the implantation.